Now student, let us come to the next question. Here the question is, which of the following is not an objective of biofortification in crops? Here the options are: option number 1, improved resistance to disease; option number 2, improve vitamin content; option number 3, improve micronutrient and mineral content; and option number 4, improve protein content. So student, which option here is the correct one? Now student, here we are talking about the biofortification. So first of all, we write that term biofortification, and we know that student, biofortification, it is used to improve the vitamin content. It is used to improve vitamin content. It is used to improve micronutrient and mineral content. It is also used to improve protein content. So student, here we have to find which of the following is not an objective of biofortification in crops. So here correct answer, improve resistance to disease, is not an objective of biofortification in crops. So student, here correct answer, it is option number 1.
Hello students, let's have a look toward the question. The question says what? Read the following statement. We have the following statement and we have to read it. The first statement, metagenesis is observed in helminthes. Do you know what do you mean by metagenesis? The metagenesis means what? The organism having the two forms, the one is named as polyp and another one is named as medusa. These forms are found in the organism. That property which is having the both polyp and medusa, that is named as metagenesis. So this property is found in the phylum Coelenterata, right? It is found in Coelenterata. So the option first is incorrect. The next one is echinodermata are triploblastic and coelomate animals. It is correct statement that echinodermata having all three layers, ectoderm, endoderm and mesoderm, with a true body cavity which is named as coelom. The next question is, roundworm have organ system level of organization of body. Organization, clear? The roundworm belongs from what? It belongs from Aschelminthes. Correct? It belongs from the Aschelminthes, and Aschelminthes having organ system level of organization. So this statement is also correct. The next statement is, complates are present in ctenophores. It is correct. When you are looking at the diagram of ctenophores, it is just like this, and this is the complates having the cilia which helps in digestion. It is correctly incorrect statement because it doesn't help in the digestion. It helps in the locomotion. It helps in the movement of the particle. So this statement is also incorrect. Till here it is correct, but it is incorrect one. The next, water vascular system, it is a characteristic of echinoderm. Yes, as we know, the echinoderm don't have a respiratory, excretory and other system. All the system are performed by a single system that is named as the water vascular system. So look at the options and find out the correct answer from the given below option. So option B is correct, option C is correct, and option E is correct. So let's have a look toward it. B, C and E. The option B, the option C and the option E are correct. So the answer goes to what? Answer goes to the third. Copy down. Good question. Hello students, let's have a look toward the question. The question is, sphincter of Oddi is present at, it is a very pretty question of NEET 2021, direct line from your NCERT digestion and absorption chapter. The chapter named as digestion and absorption, having a direct line of this. Do you know what? Do you know there is a duct which is coming from a liver or from a gallbladder? The liver and the gallbladder, there is a duct. That duct is named as what? This duct is named as the hepatic duct, correct? This named as the hepatic duct, and the another duct which is coming from a pancreas, that is named as what? It is named as pancreatic duct. This duct is named as the pancreatic duct. Both combines and forms hepatopancreatic duct. Hepatopancreatic duct. This duct is opens where? It opens in the small intestine first part, duodenum, and it is regulated by an sphincter that is named as sphincter of Oddi. So look at the options, very best question. Junction of hepatopancreatic duct and duodenum is the correct answer of this question, not of the gastroesophageal junction, not of the jejunum and duodenum, not of the ileocecal junction. The correct answer for your question goes to what? It goes to first. Copy down.
Now student, let us come to the next question. Here the question is, which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? Here the options are option number 1 zygote, option number 2 dikinesis, option number 3 pectine, and option number 4 leptotene. So student, which option here is the correct one? We know that student, after the process of crossing over is completed, it is completed in the pectine, then diplotene, and then it is in dikinesis. So student, the terminalization of the chiasmata is the distinctive feature. The terminalization of chiasmata is the distinctive feature of dikinesis. Which is the last stage of prophase 1. We know that student, the terminalization of the chiasmata, it is the distinctive feature of the dikinesis, which is the last stage of the prophase 1 of meiosis 1. So student, here correct answer, it is option number 2 dikinesis. Now student, let us come to the next question. Here the question is, the fruit fly has 8 chromosomes 2n in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, what would be the number of chromosomes after S phase? Student, here the options are option number 1 16, option number 2 4, option number 3 32, and option number 4 8. So student, which option here is the correct one? Now student, if we are talking about the mitosis in the number of the chromosome at G1 phase, so here in G1 phase, the number of the chromosome, it is 8. Now, what would be the number of the chromosome at S phase? So we have to know what is the number of the chromosome at S phase. We know that student, in S phase, the replication of the DNA occur. The replication of DNA occur in S phase. The replication of DNA occur so that amount of DNA get doubles, but the number of chromosome remains constant. But the number of chromosome remain constant. So if the number of the chromosome at the G1 phase is 8, then the number of the chromosome at S phase would also remains constant. It is 8. So student, here correct answer, it is option number 4, 8.
Over to our next question, that is, for effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Which of the following molecular diagnostic technique is very useful for early detection? Now what they are asking you, that early detection means the primary test you are performing on a particular patient to know what is actually the disease. Then you will confirm that disease by different type of procedures we are having. But the primary test you are performing by looking forward the symptoms of that patient, what are that? Let's move on to the option. First option, Southern blotting technique. Second, ELISA technique. Third, hybridization technique. And fourth is Western blotting technique. So the correct answer for this question is second option, that is ELISA technique. Why is it so? Because ELISA technique is enzyme linked immunosorbent assay. That means here the antigen antibody reaction occurs. So that is the primary stage. Once you get the symptom of the patient, we get to know that we performed ELISA test out of the 4 options I'm talking about. Okay, so out of these 4 options, the early detection is by ELISA, the antigen antibody detection. If you don't get anything, or if you get something over there, to confirm it, we can go further for any these type of hybridization technique or blotting technique. But for the early diagnostic, we have to go with the ELISA test, not with the other 3 options, that is the Southern blotting, hybridization technique and Western blotting. So that's why the correct answer for this question is option number 2. Over to our next question, that is, with regard to insulin, choose correct options. So here these statements are given over here and we have to find out that which statements are correct. Let's see. A, C-peptide is not present in mature insulin. Yes, this is a correct statement. B, the insulin produced by recombinant DNA technology has C-peptide. No, it is having only 2 peptide chain, A and B peptide. So this statement will be or is incorrect. C, the proinsulin has C-peptide. Yes, proinsulin is the immature insulin which is having 3 peptide chain, A, B and C. And when it gets mature, when it convert into a mature insulin, it will be having only A and B insulin, B peptide, C peptide is removed from there. So that's why C option, C statement is also a correct statement. D, a peptide, A peptide and B peptide of insulin are interconnected by disulfide bridges. So that is also correct. You have seen like this is A peptide and B peptide and they are connected by disulfide linkages. So this statement is also correct regarding insulin. So A, C and D statements are correct, B is only incorrect. Let's move on to the options. 1, B and C only. Second, A, C and D only. Third, A and D only. Fourth, B and D only. So you can directly take out the correct answer, and the correct answer is second option, that is A, C and D only are the correct statement regarding insulin. Thank you.
Hello students, let's have a look toward the question. Match the list first to the list second. We have the 2 columns, one is named as list first, second is list second. The option A is Aspergillus niger, Acinetobacter aceti, Clostridium botulinum and the Lactobacillus. The list second is acetic acid, lactic acid, citric acid and butyric acid. The question is from the 12th class microbes chapter. Here we have some organisms and here we have the secretion. So let's have a look. When we are talking about what? We have a learn from a lactobacillus. from mid of that, we all know that LAB, lactoacidus bacillus, it always secrete an acid that is named as lactic acid. It is responsible for the formation of curd. So you always remember, let's match D to the second, and let's have a look toward the option, not this one, not this one, only the single option is D, having the second one. Let's have a look toward the other one, the A to the power third, the Aspergillus niger, it secretes citric acid, it is also the correct one. The Acetobacter aceti, it secretes the acetic acid, it is also correct one. The Clostridium botulicum, it secretes the butyric acid, it is also correct one. So the correct answer, students, goes to what? So the correct answer of this question goes to the fourth. Clear? Copy down. Hello students, let's have a look toward the question. Match the list first to the list second. List 1 we have metamerism, canal system, complex, nidoblast. A very interesting question from a chapter Animal Kingdom. As we all know about the animal diversity, animal kingdom, we have to remember the property of the phylums. So metamerism is what? The metamerism is the divisions, and these divisions have started from a phylum named as Annelida. When we are coming to the canal system, as we all know, the poriferan don't have the respiratory, excretory, vascular or any other type of system is found in porifera. All the systems are absent. The single system which is found in porifera is named as the canal system. The complex, do you know there is a structure of the ctenophores? When you look toward the ctenophores, this ctenophores having this plates having a projection named as what? It is named as the complex. So the complex is found in the ctenophores and the nidoblast. That's why the nidoblast has, due to this, there is a name of a phylum that is named as Cnidaria, which is also known as Coelenterata. Great question of NEET 2021 from Animal Kingdom. Let's have a match. Metamerism, Annelida, canal system, porifera, complex, ctenophora, nidoblast, coelentrates. Let's have a look. A to the third we have only in the two option, rest both are incorrect. Canal system in porifera, B to the fourth in both. Next option, C, complex in ctenophores, C to the second and C to the second and only in the second option. So your correct answer goes to what? The correct answer for your question goes to the second one. Copy down.
Over to our next question, that is, if adenine makes 30% of the DNA molecule, what will be the percentage of thymine, guanine and cytosine in it? Let's see. So we are having adenine 30%. And we know that according to the Chargraph's rule, we know that the number of adenine is equal to the number of thymine, because adenine binds with thymine while cytosine binds with guanine. So adenine binds. So if you are having 30% of adenine, that means there will be 30% thymine, because adenine is binding with thymine only. So thymine will also have 30%. So we can say that AT is your 60%. Because we are talking about percent and a percent contains 100. So 100 minus 60 will gives you 40. So remaining 40% will definitely is your cytosine and guanine, because cytosine bond with guanine. Okay, so cytosine will be 20%. We are splitting this 40% equally into cytosine and guanine. So cytosine will also be 20% and guanine will also be 20%, or by Chargraph rule you can take out the answer, but apart of that, this is the correct way and the accurate way to take out the correct answer. Now let's see the option. First option, thymine is 20, guanine 20, cytosine 30, incorrect. Second, thymine is 30, guanine is 20, cytosine is 20, so this is correct. Third option, thymine is 20%, guanine 25 and cytosine is 25, incorrect. Fourth option, thymine 20% given in 30% and cytosine is 20%. So the correct answer you can see is given over here in the second option. Thank you.
Hello students, let's have a look toward the question. Select the favorable conditions required for the formation of oxyhemoglobin curve at alveoli. The question is from breathing and exchange of gases. Breathing and exchange of gases, having a good question from oxyhemoglobin dissociation curve, right? It is oxyhemoglobin dissociation curve. The question is asking oxyhemoglobin at alveoli level. Do you know what is happening at the alveoli level? At the alveoli, we know the partial pressure of oxygen is always high. The partial pressure of carbon dioxide is low because we have the oxygen in the alveoli. If the PCO2 is low, then the H plus concentration is also low and the temperature is also low. So what happened in that? At the favorable condition required for the oxyhemoglobin dissociation at the alveoli is low PO2, no. It's high PO2 but not high PCO2. It's low PO2, no. Only the last option, high PO2, low PCO2, less H plus, lower temperature. They are the correct and the favorable condition for oxyhemoglobin dissociation, because we know the binding capacity of hemoglobin with the oxygen is a maximum at the alveolar level and the minimum at the tissue level. So the correct answer of your question goes to 4. Clear? Copy down.
Now student, let us come to the next question. Here the question is, the organelles that are included in the endomembrane system are. Here the options are, option number 1, endoplasmic reticulum, Golgi complex, lysosome and vacuole. Option number 2, Golgi complex, mitochondria, ribosomes and lysosomes. Option number 3, Golgi complex, endoplasmic reticulum, mitochondria and lysosomes. And option number 4, endoplasmic reticulum, mitochondria, ribosomes and lysosomes. So which option here is the correct one? Now student, here we are talking about the endomembrane system. Endomembrane system includes, it should includes endoplasmic reticulum, that is ER. It should include endoplasmic reticulum. It should include Golgi complex. It should include lysosomes and it should include vacuoles. We know that student endomembrane system included the organelles like endoplasmic reticulum, Golgi complex, lysosome and vacuole. So here we find our answer. It is option number 1. It is correct one. So here correct answer student, it is option 1.
Hello students, let's have a look toward the question. Match the following question. The list 1 we have Physalia, Limulus and Cyclostoma punctata. A good question from an animal kingdom. Let's have a look toward the question named as animal kingdom, and we know about the Physalia. The Physalia, do you know the common name? It is also named as Portuguese man of war. It is also known as Portuguese man of war. The next option we have Limulus. It is also named as king crab. The Limulus is also named as living fossil. The reason the fossil which are living, that is named as living fossil. The next one is named as Encyclostoma. Encyclostoma look like a hook, that's why it is named as hookworm. And the last one punctata. The punctata is also named as pearl oyster. Correct? Let's have a match. The Physalia, A to the power Portuguese man of war, Limulus living fossil, Encyclostoma hookworm, punctata pearl oyster. A to the second in this option and this option. Rest both are incorrect. The B option Limulus to the third in both. The next, encyclostoma is hookworm. C to the fourth. C to the fourth in this and D to the first in this. So the correct answer for your question goes to the second, that the Physalia to the second Portuguese man of war. B to the third, Limulus is the living fossil. C to the encyclostoma is the hookworm, and the D to the first, Pinctada, is named as pearl oyster. Correct answer is second. Copy down. Hello students, let's have a look toward the question. The partial pressure in mmHg of oxygen and carbon dioxide at alveoli, the site of diffusion are. A very good question from a chapter named as breathing and exchange of gases. Breathing and exchange of gases. Let's have a look. It is asking about the partial pressure. The partial pressure in the environment is what? It is 159 mm of Hg of the oxygen. Whenever it enters into the alveoli, its partial pressure decreases to 104 mm of Hg, and carbon dioxide partial pressure increases to 40 mm of Hg. So let's have a look toward the correct option. Does 40? No. 95? No. 159? No. Because the question is asking about the site of diffusion at the alveoli, not at the atmosphere. So at the alveoli it is 104 and 40. So the correct answer for your question goes to the fourth one. Clear? Thank you.
Next question is match list first with list second. Question from reproductive health. Okay. A. Waltz, B. Intrauterine device, C. Vasectomy, and D. Tubectomy. Okay. We also know what is vasectomy and what is tubectomy. It is famous question. Okay. Vasectomy. हमने कहा we know vasectomy means removal of vas deferens, and tubectomy removal of tube. T for tube, so removal of fallopian tube. Okay, so C second and D fourth. Okay, see the options, C second and D fourth. Okay, A. Waltz first, entry of a sperm through cervix is blocked. Okay, upper the cervix. Intrauterine device, phagocytic of sperm within the uterus. Done, bacha? So correct answer is first. Okay.
Now student, let us come to the next question. Here the question is, during the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, the which of the following step of PCR will be affected first? Here the options are option number 1 extension, option number 2 denaturation, option number 3 ligation, and option number 4 annealing. So student, which option here is the correct one? We know that student, what will happen during the process of gene amplification using PCR? Gene amplification. If we are talking about the gene amplification process in which the PCR is using gene amplification. So gene amplification using PCR, if very high temperature is not maintained, if very high temperature in the beginning, if very high temperature in the beginning is not maintained, it will affected, which of the following steps of PCR will be affected first? So student, we know that during the gene amplification using the PCR, if very high temperature in the beginning is not maintained, the step of PCR is polymerase chain reaction. The step of PCR which will be affected first, It is the denaturation. It is the denaturation. So student, in the gene amplification, while using the PCR, if very high temperature is not maintained in the beginning, the step of the PCR which will be affected first, it is the denaturation. So student, here correct answer, it is option number 2. Now student, let us come to the next question. Here the question is, identify the incorrect pair. Option number 1, toxin, aberrine. Option number 2, lectins, concanavalin A. Option number 3, drugs, ricin. And option number 4, alkaloids, codeine. So student, here which option is the correct one? Now if we are moving to the option number 1, toxin, example aberrine, it is correct one. Option number 2, lectins, it is example concanavalin A, it is also been correct one. Drugs, it is ricin, it is incorrect one, because we know that student, if we are talking about the drugs, example of the drugs, it is come from vinblastine and curcumin. All of these are secondary metabolites. All of these one are the example of the secondary metabolites. But option number 3, drugs, is incorrect, because example of the drugs, it is vinblastine, curcumin. So this option number 3 is incorrect. Option number 4, alkaloids, codeine and morphine, both are the example of alkaloids. So this is also being correct one. Here we have to pick out the incorrect one. So option number 3 is incorrect answer.
Next question, chronic autoimmune disorder affecting neuromuscular junction. ओके यानी कि हमने कहा एक क्रॉनिक डिसऑर्डर है और न्यूरोमस्कुलर जंक्शन को क्या कर रहा है बेटा, अफेक्ट कर रहा है. फैटिक है, वीकनेस है, पैरालाइसिस ऑफ स्केलेटल मसल्स. समझ में कहां से आता है? पैरालाइसिस है किसका? स्केलेटल मसल्स का. तो बच्चों जब भी ऐसी बात होती है हम सीधा कहां जाते हैं? ग्रेविस डिजीज पर. और आंसर क्या आएगा? माइस्थीनिया ग्रेविस. तो जैसे सिम्टम्स है उसके अकॉर्डिंग तो इसका जो आंसर होना चाहिए वो क्या होना चाहिए? सेकंड. चलते हैं नेक्स्ट क्वेश्चन पर. Next question is, which enzyme is responsible for the conversion of inactive fibrinogen to fibrin? क्वेश्चन कहां से है? ब्लड क्लॉटिंग से. और ब्लड क्लॉटिंग की बात करें हमें पता है इसकी जो थर्ड स्टेप है उससे डायरेक्ट क्वेश्चन है. क्या पूछ रहा है? कन्वर्जन कर पूरा किसका? फाइब्रिनोजन का. किसमें? फाइब्रीन में. जब भी फाइब्रिनोजन का फाइब्रीन में कन्वर्जन होता है तो सबसे पहले क्या होता है बेटा कि जो एंजाइम होता है प्रो एंजाइम थ्रोम्बोकाइनेज, वो प्रोथ्रोम्बिन को थ्रोम्बिन में कन्वर्ट करता है. यानी कि प्रोथ्रोम्बिन कन्वर्ट हो गया किसमें? थ्रोम्बिन में. और थ्रोम्बिन क्या करेगा? इस फाइब्रिनोजन को फाइब्रिन में कन्वर्ट कर देगा. तो इसका जो करेक्ट आंसर जाएगा, एक बात ध्यान रखना जब बच्चों यहां दोनों में आपको बहुत कंफ्यूजन हुआ होगा, लेकिन उससे पहले की स्टेप ध्यान से पढ़ो. वो पूछ रहा है कन्वर्जन. थ्रोम्बोकाइनेज हमने कहा एक्टिव करने का काम है, लेकिन कन्वर्जन किसने किया? थ्रोम्बिन ने. तो इसका जो राइट आंसर जाएगा वो क्या जाएगा? थ्रोम्बिन. कहां से है क्वेश्चन? सर्कुलेशन से, ब्लड क्लॉटिंग से. डन बच्चों, हो जाएगा. बहुत इजी क्वेश्चन है. क्या आएगा? थ्रोम्बिन. क्योंकि थ्रोम्बिन क्या करता है? फाइब्रिनोजन को कन्वर्ट कर देता है किस में? फाइब्रिन में. ओके.
Now student, let us come to the next question. Here the question is, the centriole undergoes duplication during. Here the option number 1, prophase, option number 2, metaphase, option number 3, G2 phase, and option number 4, S phase. So student, here which option is the correct one? We know that student, in cell cycle, the centriole gets duplicate during the S phase, of the interphase. So student, where the centriole gets duplicated. So student, here correct answer, it is option number 4, S phase.